Well, welcome to all of those of you here and to all of you on Zoom. Thank you for coming to the second installment of our Journey seminar series. So we're hoping to give you guys a little taste of not just what research is all about, but what getting into research is all about and the bumps and twists and turns in the road along the way. So we're very excited to have back with us today a DePaul alumni for Blue, not only for DePaul Blue, but also for Go Blue, because she's currently a postdoc at the University of Michigan. So Ramiah Jacks got her BS in biology at DePaul in 2013, and then went on to get a PhD in immunology at Loyola in 2019. And like I said, is currently at University of Michigan and is interested in interactions between obesity and immune function, specifically looking at interactions between T cells and the extracellular matrix and adipocytes. So I'll let her fill in the details of that and also her experiences at DePaul and beyond. But again, thank you all for joining us today. And I will have the chat open myself. So if you guys have questions as we're going along, we'll probably save most of those to the end. But feel free for those of you on Zoom to type questions in the chat, and those of you who are here can go ahead and ask questions in person once Ramiah's finished her talk.
Great, thank you so much for that introduction and the invitation. Thank you all for coming on Zoom, hi, and in person. I'm so excited to be back on campus. I graduated in 2013, so being back here for one of the first times and since I don't want to calculate the number of years. It's just so exciting and so great. So I love coming back here. Thank you again for the invitation. Today, I'm going to talk to you a little bit about my journey, what I've been up to since graduating from DePaul. And what I hope to accomplish today is to tell you a little bit about my postdoctoral experience and then get into a little bit of my journey. How did I end up to where I am now? And when I'm talking about my journey, I'm going to sprinkle in some of my advice that really helped me on my journey, and hopefully it'll be helpful to you as well. So I'm first going to start with, well, what do I do now? So after I finished graduate school, I knew that I wanted to do a postdoc. And a postdoc is a position that you can take after graduate school to get more experience doing research, specifically doing more independent research, because sometimes in graduate school, well, most of the time, in graduate school, your research is very mentored. So your postdoc is the time where you get to figure out what exactly you want to do and gain that independence in research. But I also knew that I wanted to teach eventually. So I wanted to find a program or a position in which I could do both. So the University of Michigan has what's called an IRACTA program, and IRACTA is an NIH grant in which funds individuals who want to go into academia to do research and teach. And IRACTA is an acronym that stands for Institutional Research and Academic Career Development Award. So I am in the IRACTA program at the University of Michigan. And in this program, I'm able to do meaningful research, which I'll touch on in this presentation. And I'm also able to have mentor teaching experiences as well as independent teaching experiences and learn more about best practices for teaching. So I'll touch a little bit about what my day-to-day life looks like in the beginning of this talk. So first, let's talk about what my research interests are. My research goal, specifically working with Dr. Carrie Luming at the University of Michigan, is to understand adipose tissue inflammation in the obese state. So first of all, what is the obese state? Well, obesity is defined as a body mass index or BMI of over 30. And I put this little asterisk here to remind myself to tell you that we acknowledge that the BMI, which is measured by your body weight divided by your height squared, isn't the best measure of assessing body fat. So we know that somebody with could have a high BMI, but that could mean that they have a high lean mass and not a high fat mass. They might be very muscular, but their BMI would still be high. So we acknowledge that obesity can't directly be pinpointed to the BMI number. However, right now that's like the gold standard of the definition of obesity. There are other measurements by which we can use to more accurately define the composition of somebody's body fat. One of those is a bod pod, which is like this egg-shaped instrument in which a person can sit in, and it can measure their weight and their body density and calculate their body fat composition. And obviously that's like much more invasive than just calculating a BMI. So the field typically uses BMI as the gold standard to set a threshold for obesity. So let's talk about the background of obesity right here in the United States. What I want you to learn from this slide is that if we look at this map of the United States, we can see that as the years go on, there's a change in the map. And most drastically, you can see that the green states are states that where the prevalence of obesity in adults is from 20 to less than 25%. However, the red states, the prevalence of obesity in adults is greater than 35%. And what you can appreciate is that as time goes on from 2011 to 2019, we're starting to see a very rapid increase in the number of states that are moving from that green, yellow to the more red designation, indicating that more states are having a greater prevalence of adults with obesity over time. And even more interestingly, if we look at the map for 2020, we can see that almost all of the states are red, and there's the emergence of this even darker red color. The CDC had to update their legend to include states that are greater than 40% prevalence of obesity in adults in the United States, and even project that in future years like 2021, 2020, the number of states in which the obesity prevalence is increasingly greater is going to continue to grow. So obesity is certainly very prevalent in the United States. And while we know that as a fact, why do we care? Why should we care that the number of adults who are obese is increasing so rapidly in the United States? Well, we care because there are a number of health complications that are associated with obesity. This is just a small list of some of the associations that individuals are studying currently. These include things like high blood pressure, coronary heart disease, stroke, osteoarthritis, many types of cancers. You might be also familiar with type 2 diabetes, which refers to an individual not being able to respond to insulin and use glucose for energy as efficiently. But however, because I'm an immunologist, I'm really interested in the fact that we observe an impaired immune function in individuals with obesity. What do I mean by that? Well, we know that in individuals with obesity, there is a inhibition or lack of ability for the cells in their immune system to respond to infection. So we can appreciate that the immune system is responsible for fighting off infection, protecting us from viruses and bacteria and things like that. However, in individuals with obesity, their immune system doesn't function properly. It's actually very dysregulated and doesn't provide that protection against pathogens that we expect the immune system to. And further evidence for this was really highlighted during this COVID-19 pandemic. The CDC reported that obesity increases the risk for severe COVID-19 associated illnesses. This is directly related to the issue of people with obesity having this inability to fight off viruses compared to lean individuals. Further, it's been known in the field for a few years now that individuals with obesity also have an altered response to vaccinations, especially the flu vaccination. So it's been shown in this study from Melinda Beck's lab in 2017 that individuals with obesity have an increased risk of contracting influenza even after vaccination. And they discovered that the reason why individuals who are obese and were vaccinated against the flu were not protected from the flu was because they have an impairment in the cells in their body called T cells, which also are required for fighting off infections. So there's something different, dysfunctional about T cells in individuals with obesity that leads to them not being able to mount the proper responses after flu vaccination to be able to elicit effective protection against influenza. So because we know that obesity is prevalent and we know that obesity is associated with immune system dysregulation and dysfunction and impairment, our lab really wanted to understand, well, what is happening in the obese state that's leading to these outcomes that we are observing? So in order to answer that question, our lab really wanted to focus on looking directly at the adipose tissue itself. So adipose tissue is a fancy word for fat. And the function of fat is to store excess calories and nutrients. So what I'm showing you here is just the cartoon depiction of lean adipose tissue. So in the lean, healthy state, your adipocytes, which are your fat cells, function to store lipids. The extra calories that you don't burn off during the day, your body will store those as lipids. So in a time of nutrient deficiency, maybe when you're fasting, your body can break down those lipids stored within the adipocytes and use them as energy. However, in the case of overnutrition, such as in the case of obesity, your adipocytes have an influx of lipids that need to be stored. And so in order to accommodate this great increase of lipids that now need to be stored because of this overnutrition, your adipocytes can undergo what's called hypertrophy, which means they can grow much, much larger than their normal size. And this is actually dysfunctional. When adipocytes get this big to accommodate all of the lipid needing to be stored, they no longer can have their normal function, and that's actually very detrimental to the homeostasis of the adipose tissue itself. So adipocyte hypertrophy is one factor by which is a feature of obese adipose tissue compared to lean adipose tissue. In addition to this hypertrophy, there are also an influx of cells from our immune system that are going to the adipose tissue. Interestingly, in the lean adipose tissue, there are cells called macrophages. Macrophages are a part of our immune system that normally function to help us fight off against bacterial infections, but they also have functions in maintaining tissue homeostasis. And in the lean adipose tissue, what's called M2 macrophages are the macrophages that arise and are there to help keep everything calm, make sure there's no infiltration of immune cells, secrete small chemical mediators called cytokines to keep everything anti-inflammatory. And this is what is normally happening in the lean adipose tissue. This is healthy adipose tissue. However, in the obese state, you can see now there's these red macrophages called M1 macrophages that are starting to appear. And these are making what's called pro-inflammatory cytokines, chemical mediators that are telling other cells, hey, come here, there's an active infection, come help us resolve the infection. However, there's no infection here. This is just adipose tissue in the obese state, but it's getting inflamed and infiltrated with these immune cells. And that's very detrimental to the function of adipose tissue. It's another feature of obese adipose tissue, which makes it different from lean adipose tissue. Finally, another cell type that is in adipose tissue are called T cells. T cells are another part of our immune system that help us fight off infections. In the lean state, there are regulatory cells that help maintain the no inflammation state, help maintain everything to homeostasis, keep everything calm and everything good. So that's what's normally supposed to happen. But in the obese state, there are other T cells that are pro-inflammatory that come in and they cause more damage, secreting more cytokines, telling more cells, hey, let's fight this infection, even though there's no infection. So now we have this giant infiltration of macrophages and T cells and other cells that aren't supposed to be there, that aren't helping for homeostasis. They're actually participating in the destruction and the dysfunction of adipose tissue in the obese state. So this is something that's known in the field that is a key feature of obese adipose tissue, and that there's this giant infiltration and inflammation of immune cells that participate in the dysfunction of this tissue in the obese state. So because there's this adipose tissue inflammation in the obese state, that also leads to systemic or whole body low-grade inflammation with impaired immune system function. So because of what's happening in the adipose tissue, it's leading to effects within our entire body. Our entire immune system is now affected by what's going on in the adipose tissue. And that is leading to these impaired immune functions that we see in persons with obesity with severe COVID-19 and persons with obesity with vaccination against influenza. However, they are not protected against influenza. Also, we know that this inflammation is leading to insulin resistance because of all of these chemical mediators that are being secreted in this obese state, that leads to cells being unable to sense insulin. And that's important for being able to take up glucose and use it for energy. So this adipose tissue inflammation it's been shown in the literature to lead to and promote and be associated with the development of insulin resistance, and in humans, the further development of type 2 diabetes. So this adipose tissue inflammation is a really big problem, and we're trying to understand why are these cells infiltrating adipose tissue in the obese state? What are they doing, and how can we stop them? So in order to answer all of those questions, our lab has a number of projects trying to characterize what the macrophages are doing in the adipose tissue, as well as what the T cells are doing in the adipose tissue. But because I'm a T cell biologist from grad school, I'm a little biased. So I'm only going to talk about the T cell story today. I'm going to tell you about what we're trying to understand about the T cells in the obese adipose tissue. So we really want to characterize these T cells and understand what are they making, what are they responding to, and what are they doing in this adipose tissue? So in order to begin to answer those questions, one really important experiment that the lab did was use a mouse model of obesity, whereby you can put a mouse on what's called a high-fat diet, and that means the diet has 60% fat for 18 weeks. And what this will do is it will generate a mouse that will mimic what happens to human adipose tissue in the obese state. We can compare that to lean mice fed a normal mice chow diet, their regular mice food, and ask what are the differences in the T cells in the adipose tissue in this obese mouse compared to this lean mouse. To answer that, we can euthanize the mice and harvest their adipose tissue. And then we isolated out the T cells specifically from the adipose tissue and stimulated them in the lab in ex vivo. And we stimulated them with antibodies that will trick them into thinking they're seeing a pathogen, so we can see how they would respond. And we asked, do the T cells from lean mice respond the same or differently than T cells from obese mice? And the answer is they respond very differently, actually. We can see that T cells from lean adipose tissue upregulate surface activation markers, meaning these are markers that are known to be expressed on the surface of T cells once they're activated. And they make cytokines, those small molecule chemical mediators. And that's another feature of T cell activation. However, T cells from obese mice do not upregulate surface markers and do not make cytokines. They're actually impaired. And we actually found out they're exhausted. They no longer can respond to stimulation because they just #can't, they're done. So we don't understand why the T cells in the adipose tissue in the obese state are exhausted, but the T cells in the lean state aren't. And we have a number of hypotheses. One of the hypotheses is that the T cells in the obese state are constantly responding to something that is present in the obese adipose tissue. What that something is, we don't know. We're still trying to figure that out. But they're becoming active and they're working hard and they're trying to resolve something, and then they're exhausted. Another hypothesis is that there's something in the obese state that's being produced to suppress T cell activation. Maybe there are some molecules secreted by other immune cells in this obese state that are saying, hey, let's calm down this inflammation. Let's try to resolve this so we can get back to being healthy. And perhaps that's suppressing or inhibiting the T cells from being able to respond anymore. So we're currently doing experiments to test each hypothesis to see what's happening with these T cells in the adipose tissue specifically. So my central research question is, why is there adipose tissue T cell impairment in the obese state? What are the factors that are causing it, and can we reverse it? So in order to answer those questions, I'm using mouse models, mouse knockout models, to understand how can we improve the T cell responses in the adipose tissue and resolve this inflammation in the obese state. So that's my research. That's what I'm working on. Just a little bit, that's one project. So I'm not going to tell you the rest. I don't want to overwhelm you. So that's the research side of my job. But as I told you in the beginning, I'm also really interested in teaching. After I complete my postdoc, I want to get a job in academia, be a professor at a college. So I wanted to get some experience in teaching. And the IRACTA program is wonderful for that because, especially at the University of Michigan, we have a Center for Research on Learning and Teaching. And that center offers many courses and classes on best practices for teaching, best practices for implementing activities in the classroom to enhance student learning and retention of the information. So I was able to take a bunch of classes during this time in order to gain that information. I was also able to participate in classes and workshops and events that teach us about strategies that value diversity in the classroom, promote equity and inclusion in the classroom as well. So I'm able to take those practices and apply them when I'm teaching, which is very phenomenal because that's something that's super important to me as an educator, to be able to make the classroom a community feeling, a safe space for everyone to feel valued and respected, as well as to make students want to stay in the sciences. Because it's known that after about those first 2 years of undergraduate classes in the sciences, research has shown that undergraduates move to a different field for one reason or another, move away from the STEM fields. And so I want to be part of the solution in order to teach STEM in a way that is more accessible and such that students will feel a better sense of belonging in what can be an extremely difficult field. And so that we can increase the retention of students who stay in the STEM fields after those first couple of years of undergraduate courses. So I was able to take what I learned from the Center of Learning and Teaching at University of Michigan and actually teach at our partner community colleges. So I was able to give guest lectures in the Anatomy and Physiology course at Wayne County Community College. And I was also able to co-teach a course last summer at Henry Ford called Microbiology for the Allied Health Sciences. And that was a really fun experience because that was completely virtual over Zoom. So I co-taught the course with another IRACTA fellow, and the course was an 8-week summer course with 16 weeks worth of content. So it was a very crash course, a short amount of time, a lot of material. And so that was a very interesting first time teaching experience because I wasn't able to get that student engagement that I wanted. So I was teaching to black boxes, but that's okay. We're in a pandemic, we're living in unprecedented times. So we were able to make the best out of it. And we actually learned that the students really liked communicating with us through the chat feature. So we were able to encourage and implement that and get great student engagement and participation in the course. So that was a nice little pivot and learning experience during the pandemic. I'm also very interested, as I touched on a little bit in the previous slide, on diversity, equity, and inclusion efforts. And so in order to increase the number of minoritized, underrepresented groups in STEM, I'm working with a working group that's part of the Nutrition, Obesity, and Research Center, National Institutes of Health, Diabetes, Digestive, and Kidney Disease funded grant. That is a working group of scientists who are interested in coming up with and implementing practices that will increase the retention and the presence of underrepresented groups in STEM specifically. So this group was actually designed and implemented last year. So we're just now starting to meet more regularly, trying to brainstorm ideas and things that we think that would increase the retention of and recruitment of individuals from minoritized, underrepresented groups in science, specifically in biomedical research and obesity and diabetes research. So this was a really fun endeavor. This is an endeavor of institutions who are doing nutrition obesity research all across the United States. So it's nice to interact with individuals from different institutions, all with a common goal of trying to increase and promote and respect and celebrate diversity in research. So I'm really proud to be working with this group at the University of Michigan. So I just told you about my postdoctoral experience and what I'm doing now. So I want to jump into my journey, what I did at DePaul that was super helpful to getting me where I am now. So first off, well, how did I even know that I wanted to do research? Well, my interest peaked in research because my mother has multiple sclerosis. And if you're not familiar, multiple sclerosis is an autoimmune disease by which the body's own immune system starts attacking this myelin sheath of nerves, inhibiting the ability of the nerves to communicate effectively. And this can severely impair movement and the body's ability to effectively communicate from the brain to the rest of the body. And so when I first learned that my mom had multiple sclerosis, I was maybe 12 or 13. But I wanted to know, I was asking her all these questions like, okay, well, what can we do about it? What do scientists know? Like, well, I need more information. She was like, well, people don't know yet. Like, there's still people researching this. I'm like, research, what's that? So I went on the internet, and I don't remember if there was Google back then, but I went on the internet and I did a lot of looking at different things and looking at different papers on what people are doing and what do scientists know right now. And that was just super exciting for me. I was like, there's so much more we can learn. I am not satisfied. So I was like, okay, I need to be in this career because I want to move some things along and learn some things. So that's where I really got interested and really learned about what is research, what do scientists do, and how can we figure out the answers to all these questions that we have. So fast forward past high school, I knew I wanted to go to college, and I picked DePaul because when I came here for the, student orientation where they take you around and you talk to students and stuff, all of the students were so nice and helpful and just happy. It was just a really pleasant place to be. So I chose DePaul to do my undergraduate work at. And while I was at DePaul, I had still never had any true research experience. There was no research experience for me in high school. So I didn't really know what research looked like from a day-to-day basis. However, I was able to join the CIRIS organization, which is a Chicago initiative for research and recruitment in undergraduate science. This was a program that was here that really wanted to introduce students to all different types of research. So in order to do that, we went to the Field Museum, the Shedd Aquarium, the Argonne National Lab, and some other places around Chicago just to get us introduced to different types of research, chemistry, physics, marine biology, things like that, even plant research. So in addition to going to all these locations in Chicago and asking what scientists do on a day-to-day basis, we also did some research ourselves with collaboration from scientists across Chicago. And we did research here at DePaul, and I was actually doing plant research. We were asking, well, can we use plants to remove toxins from the soil and the water? So we set up a hydroponic situation where we grew plants in contaminated water that we contaminated in the lab, and we looked to see if the plants could remove the toxins that way. I don't remember what else we did, but I remember that. And it was a great experience. And one piece of advice here is that I actually wasn't going to join this program because, I wanted to do immunology research. I was very interested in autoimmune diseases and things like that, but they weren't, that's very difficult research to just jump into. So that obviously wasn't an option, but I'm super glad that I did join this, even though I was hesitant about doing plant research. It was such a wonderful experience to get that hands-on training and just start to acquire that scientific mind of coming up with a question, coming up with your hypothesis, how are you going to test it, what did your results look like, and what can you alter based on what the results look like from your experiment. So my advice here is to do not put yourself in a box. Like, don't be afraid to step out of your comfort zone and do things that maybe you thought you wouldn't like, because you could really end up loving it and making a lot of connections and friendships and things like that. So this was a really wonderful experience for me. In addition to doing the CIRIS undergraduate research summer program, I also joined the lab of Dr. Carrie Southern here, and this was my first immunology research experience. She is studying antibody shapes and using a technique called FRET, which we use to try to understand the shape of antibodies using fluorophores, which are chemicals that can emit light. And so I was able to be a part of her lab for probably most of the time I was here. Maybe 3 of the 4 years I was here, I was in her lab, which was really great because science isn't always up, and up. Like there are periods in which you're stagnant or writing or literature reading, and then there are periods when you're doing experiments, and there are periods when you're literature reading again. So to get that experience of really getting a true understanding of what it means to do research on a day-to-day basis by being in a lab consistently for 3 years was really phenomenal. I'm so grateful for Carrie for letting me to be in her lab and giving me phenomenal advice and a phenomenal opportunity to do research. It was really, really wonderful. So if you're curious about research, like I would certainly ask individuals if they're looking for a student. And if they are, really be serious about committing to that process and being upfront about what it is you want to learn and gain from the experience, and being committed to that experience. So this was a really phenomenal, real research experience for me when I was here at DePaul. Through that experience, I also was able to talk about my research at conferences, which is another huge part of doing science, communicating your science to other scientists. And when I was here, they had this alliance program for minority participation in science, whereby they actually paid us to do research as well as to go to the conference and talk about the research that we did. And that was really great because it took the burden off of having to find a second job and then balance that work with doing research in the lab. So this was a really phenomenal opportunity. So a little bit of advice I'm sprinkling again here now is to seek out those funding opportunities. There are actually a lot of things available if you just look for them or ask around and see what's out there, because this was a really phenomenal way for me to also interact with other people in Chicago who were part of this program, who were at other institutions, and talk about our science together at the conference. I was able to give a talk, won an award for the talk, so that was the highlight of my undergrad career. And it was also really great because you're always going to need to be presenting your science. So getting an early start on learning how to do that was really great. I really enjoyed that experience as well. And another phenomenal thing I was able to do while I was at DePaul that really helped me throughout my journey was when I was here, we had what's called the Biotechnology Club. And I became president of the club because the presidents were leaving when I came in, and they were like, hey, do you want this club? I was like, sure, let me take it over. So I took it over, and what I decided to do with it was I wanted something for individuals who wanted to go to grad school. They had the medical club, they had the veterinarian club, they had clubs for a medical centric pathway, maybe nursing, physicians, things like that. But I wasn't satisfied with what was out there for us individuals who wanted to do biomedical research. Like, how do we get into grad school? What is that process? What does it look like? What does it entail? What do I need to be doing now while I'm in undergrad to make that a smooth transition into grad school? So in order to answer all those questions, I reached out to program leaders of graduate schools around Chicago and asked them to come here to speak to us about what they are looking for in an applicant and what we could be doing at DePaul to make our applications strong and what we can expect when we enter into graduate school. And this was so helpful. I cannot understate, I can't overemphasize this enough, like reaching out to individuals who know the answers and getting that information is great. If you take away nothing from this presentation, take away, ask for advice, ask for help, ask for, how did you do it? What should I expect? What should I be doing? Don't be afraid to ask those questions. I even went to other professors here at DePaul and ask them, well, what was your journey? What do you wish you knew? How can I get to where you're being? And no one's told me, no, I don't have time to meet with you. everyone was like, oh yeah, absolutely, we can talk about this. I sat in their office and talked to them about this before I applied to grad school. So I was able to do that informational interview and get all that information before entering grad school. So I knew exactly what to expect when I entered into grad school. So ask, ask, ask. I've never had anybody tell me no. So there's that. So after getting all that wonderful advice and doing those things at DePaul, I wanted to point out another thing I did at DePaul. They had resources for students to do practice interviews, and I don't remember what those resources were called, but I did take advantage of that and did practice interviews before my grad school interviews. And that was phenomenal because I had never done an interview like that before. So to get that practice before doing the real thing and to try to kind of guess or get a better understanding of what types of questions they would ask me, how to prepare, what to expect here at DePaul at like a very low stakes situation was great, because then I was able to just sail through my interviews at grad school. So taking advantage of the resources here is another piece of advice I would say that helped me on my journey. So because of the things that I did at DePaul, I was able to make a smooth transition into grad school. And in grad school is where I really started to do that hardcore immunology research. Because at DePaul, I was looking at antibody conformations with Dr. Southern, but it wasn't true immune system immunology. I wanted to get to that point. And so I was able to get there based on the things that I did and undergraduate. And I actually did not get a master's. I just went straight from DePaul to the PhD program at Loyola. And I was able to do that because of the research experience I had here at DePaul and all of the information I gathered before I made the decision about going to grad school. So I did my graduate work with Dr. Makio Iwashima, and I focused on understanding, well, how are T cells regulated? And that's a really interesting question because T cells can do a lot of things. They're a really important cell type. I'm not biased. In our immune system, they have many functions. They can help fight off different pathogens, so viruses, bacteria, fungi, but they also can promote diseases, like in the case of autoimmune diseases. There can be very T cell driven autoimmune diseases where T cells start attacking our own body. And T cells can also help fight against things like cancers. Like there are T cells that can help prevent cancers from growing. So T cells can do a lot of different things. And I wanted to know, well, how do we know, how does the T cell know when to do one thing versus the other? And how can we control that? And so what I studied when I was in grad school is a small molecule mediator, a cytokine called transforming growth factor beta. And I studied this cytokine because it can tell the T cell to do a number of different things. It can tell the T cell to be a certain type of T cell, like a T cell that responds to bacterial infection, or a T cell that regulates other T cells. Those are very different roles, but one cytokine can tell the T cell to do those different things. And this cytokine can also tell T cells to stop being activated, stop proliferating, stop doing what you're doing, and just rest, just be. So how can one cytokine tell T cells to do this specific role? And then at the same time, the same cytokine can tell T cells to stop. I really wanted to know how. How does one cytokine have these two very different roles in T cells specifically? So in order to answer that question, I had a human T cell line in the lab, which is an immortalized T cell from humans that can live forever in culture. And so I used the CrisprCas9 genome editing system to knock out the ability for T cells to sense this TGF-beta cytokine, so they can no longer see it or sense it or respond to it. And I asked what happens in those cells and was able to find a really novel and interesting signaling pathway that people didn't know that TGF-beta and T cell signaling could intertwine in this way. And so by using CRISPR-Cas, I was able to identify the signaling pathway and identify molecules that are now involved in TGF-beta signaling and T-cell receptor signaling. So that was a really amazing experience. And also, when I was doing CRISPR-Cas, like now it's like super, everybody knows about it. But when I was doing it back in 2013, it was so new. Like I was reading some of the first papers published about it, and some of the first papers trying to look at humanizing the Cas9 to make it more efficient in human cells. And so doing this project was rough, but it was such a great learning experience to be able to ask a question and use these novel new techniques to identify something that the field hadn't seen before. It was such a rewarding experience, and it really affirmed my desire to stay in research. And that's why I wanted to do a postdoc after grad school, because I really liked that experience of asking questions and discovery. And what I also really like is community outreach. So while I was in grad school, I also participated once a week in what's called Science Club. And that was a club that was at a Boys and Girls Club in Chicago that brought in 4th, 5th, and 6th graders from a very underserved area in which their science and math skills, as assessed by things like state test metrics, was severely lagging behind compared to other communities. And so we wanted to give them a more positive science experience. We wanted to show all of the students that becoming a scientist just means to ask questions and figure out how to answer them. And a scientist can look like anyone. And so what we did is we did fun little experiments. In this example, they're eating ice cream that they made themselves using liquid nitrogen, which was really fun. And we tested different milks like almond milk or high fat milk or skim milk to try to see which ice cream would taste the best. Obviously, the more fat, the better when it comes to making ice cream. And so they learned that, that's science. And so it was a really fun way to get students to be like, oh, okay, this is science. I didn't know this was science too. This is actually pretty fun. So our hope in doing Science Club is that these students will remember these experiences and interactions and maybe thinking about obtaining their own careers in science when they go to middle school, high school, and beyond. So during my journey, what would be my advice? So I already sprinkled some in of my biggest advice, and that would be to ask questions and seek advice. Whether that be to, if you're specifically interested in going into a program, maybe ask people that are in that program, what is it like? Or ask the director, or ask individuals who are in the job that you want, how did they get there? Attend career fairs, talk to the people doing your goal job, join a summer research program, because that was a great way to be exposed to a broad range of different types of research. And my final piece of advice is to follow your passions. So all throughout undergrad, grad school, and even now in my postdoc, I'm still doing the things that I love, which one of those things is to find and promote ways in which to increase the number and representation of minoritized, underserved individuals, groups in STEM. So I love participating in activities that also align with those passions, like community outreach or this DEI task force that I'm currently a part of. And that also includes doing things like crafts or going to the movies or things outside of the lab. Science, yes, that's a huge part of my life, but I'm also a human being who does other things outside of the lab. So in order to keep you sane and keep the balance, I also, another piece of advice is to still do other things that make you happy. You don't have to be in the lab 12 hours a day. You can go outside and take a walk or make a painting or whatever it is that makes you happy and brings you peace. My advice is to continue to do those things and know that sometimes it does get extremely difficult in science, but it's worth it. And you have a community of individuals who you can lean on. You can ask for advice and ask for help and ask for support. So with that, I'll take any questions, and thank you so much for your attention and time.
And again, if anyone on Zoom has questions, you can go ahead and type those in the chat and I'll relay those to Dr. Jacks. Yes.
So you ended there talking about work-life balance. Was that something you always had in place all the way through, the kind of balance of, outside of work, outside of school, or is it something over time you had to appreciate more the importance of work-life balance?
Certainly the latter. Definitely, I had to appreciate it more. Do I need to repeat the question? Do I need to repeat the question for Zoom, or they can hear? They can hear. They can probably hear it. Yeah. Okay.
Yeah.
Okay. Awesome.
Thank you, sir.
Awesome. Yeah. So, especially in grad school gets so busy.
Back and forth there says it all.
Yeah. and that's why I was really glad I found Science Club. And university or Northwestern was emailing everyone about getting grad students to participate in Science Club. When I saw the email, I was like, this sounds like a nice break from lab. So I was really glad that I saw that email and joined and got to do some fun things and get out the lab for a little bit. And that also helps to, when you step away from it a little bit, you can think a little bit more clearly. Like I learned that too when I go for a long drive. I can think about it a lot more clear than if I'm just sitting at my desk staring at paper reading it for the 10th time. So I definitely had to appreciate to smell the roses after grad school and during grad school.
So that's a challenge, right?
Yes.
You figured it out later on. I think most people, you and before you, have figured out later on is how to get it earlier. Right?
Yeah.
How to get that message across earlier. Like
Yeah.
first year classes, like do you have a suggestion on that? What point that as you're working with groups, that kind of notion of balance being introduced earlier? Thoughts on that?
Yeah, that's a great question. So what I think about is when I have I don't like thinking about the future, but having lab group outings, like taking a break from the laboratory and going out when we can to lunch or the park or things like that, to like build community, but also as a little refreshing moment away from science and just get to know people as they are as human beings, like have that human being connection, which I think even after the pandemic, people in general are more appreciative of your time and your value as a human and not just as a worker. So I think and I hope that's going to continue on. But I would say like in the undergraduate stage, like build community with your lab mates, go on lab lunches or do other things on your own that give you a chance to escape lab for a little bit and foster your peace. Yes.
I have a question. So what would be your advice of when facing like rejections or failures like in grad school or something?
Excellent question. So I'll start with the failure part first. So I think failure is necessary. However, I don't think it defines you. So I'm pretty sure all the professors in this room have failed, but what is your definition of failure and what do you do after that? And so there are certainly times in which you submit your paper, but the journal didn't accept it. What are you going to do? Just stop working? No, you're going to submit it to a different journal. You're going to do the revisions to make it so that it can be accepted to a different journal. And so what I like to do is use my failures as a learning experience and you're not a failure, you just failed. That's a big difference. So, and then the other part of your question was?
Like facing rejections for like grad school or something.
Yeah, rejections for grad school, yeah, that one stings. So, but just because one school didn't want you doesn't mean another school wouldn't. I was rejected from Northwestern but accepted into Loyola, and so that didn't stop me. And so, yeah, in life, rejection is a part of life, but just frame your mindset such that if you do your best and you're proud of what you accomplished and how you answered the questions and where you are in the stage of your life, then don't let that define you. Just allow it to be a learning experience.
Going off of like with that, like how did you keep yourself like after you face like a rejection? How do you keep yourself motivated to keep going? Like that's kind of hard. Like what is your advice to do stuff? I don't know.
Yeah, so One of my friends told me that her PI told her that it's okay to, you know, cry, eat the ice cream that night, but the next day, get up and work. So I take from that is it's okay. Your feelings and emotions about the rejection are valid, and it's okay to acknowledge them, have some time to reflect on that. And then when you're finished with that, use that as motivation. Okay, what are you going to change such that you don't have this experience again? So that would be my advice.
And you have the other thing there, right? Follow your passion. So when you have your passion there, it's kind of easier to pick yourself back up, right? When you follow your
Absolutely.
passion versus
Yes.
something else.
Yes, thank you.
Do you have any other questions from the crew on Zoom?
I have lots of questions, right?
We're also going to have a reception in Miguel North afterwards too. But
I just want to ask this out loud because sometimes, speaking for myself and for other students that I know, asking just right now is difficult because you really want to reach out, but you don't. Sometimes you don't find the words of how to do it or the way to proceed in general. But do you have like, and I know there's no recipe or something like that, but any suggestions for in general for students, like how to reach out? What's the best? Because you just gave that advice. It's clear, just reach out. But do you have any suggestions, advices additional to that?
Yeah, so what I did is I emailed professors of whom I took their course, so I knew them a little bit and they knew me a little bit, so that was a little bit of an icebreaker. I wasn't a complete stranger. I also solely emailed in the beginning because I was not comfortable asking in person after class. So I sent an email to them and allowed them to respond via email so then we could talk one-on-one at the time of their choosing. But yeah, I emailed individuals of whom that I took their class. Also ask around your peers and see who they've talked to, because they could maybe give you information on individuals who are more receptive to that, which again, I haven't had anybody tell me no. So I don't think you're gonna have that problem here at DePaul. But yeah, asking your peers too to see if they've done something similar, what they learned from that experience and how they navigated that as well. But yeah, I certainly emailed professors after class like, hey, I maybe want to do your job one day. Could you give me some advice?
Flattery never hurts.
Yeah.
So it comes up all the time in discussions. It's kind of varies based on different thoughts on things. But notion of success. What is success? Like, what is professional success? It's a big question, right?
Yeah.
Because it's the obvious go-to. Like, I did this, I got my PhD, right? Is that something you think about much or thought about much, or are you talking to other students to think about what success means? Is that something you think about?
It is not something I think about at all, actually, because even though I've accomplished the things I've accomplished, I'm never like, oh yeah, I'm awesome. I'm still always trying to reach my next point in my path. Like I'm still very much, yeah, I've accomplished a lot, but I have a lot more to go. Like there's a lot of things I want to check off my list. And so how I think about being successful is, how do you feel after you've accomplished that? Do you feel that you gave your all? Are you proud of what you've done? That's what I value more than, well, how many papers did you get, or what university did you go to get your PhD? I hope to instill that individuals want to do their best to work at all times, because that's the way in which I measure somebody's passion or interest or success in their particular field is, how are you putting in the work, and are you proud of the work that you're doing? And if you're proud of it, then I'm ecstatic. And so that's how I think about it when I do think about it.
Yeah, I thought about giving an amazing answer. So
I don't want to cut off this conversation. I want it to continue, but we also promised that Dr. Jacks will be available in the Beyond Our Patriots and chat with students afterwards. So just in case some were unable to make the seminar but want to join for that conversation, I would encourage all of you guys to move that way as well. There will be snacks. And you also gave me a great segue into how I wanted to finish. And that I'm sure many of you have already heard the quote, success is where opportunity meets preparation. And I think Dr. Jacks did a great job illustrating that, because if you just look at your resume, you would say, wow, you've been incredibly successful and here are all these things you've accomplished. But by sharing your story, you can see how much preparation went into those accomplishments. That when you got an opportunity, you had already investigated it, you already had those conversations, you were open to things that you hadn't necessarily thought about immediately, but once you learned more about them, the value of that opportunity presented itself to you. So I think that you're a great illustration of that quote, and I really appreciate you sharing the ins and outs of your story as well as the work that you're doing today.
Thank you for having me.